This video is about the Gram stain lab. Gram staining is a procedure that's super simple. We would normally do it in class in just one period. And the idea behind Gram staining is that it allows us to classify bacteria into one of two main groups. When you Gram stain bacteria, it does not tell you the exact species of the organism. Of course, there are thousands and thousands of different types of bacteria species, but rather Gram staining tells us which of two groups it falls into. Is it Gram negative or is it Gram positive? Those two groups are different based on their cell membranes. To remind you of freshman bio, a cell membrane is something that goes around the outside of a cell. It keeps the environment outside of a cell separate from the internal environment of a cell. A cell uses its cell membrane to regulate what comes in and outside of it. You can kind of think about it a little bit like your skin, where it's a barrier difference between what's inside versus what's outside. However, it's a little more open, permeable, and lets things through more so than like your skin does. We will spend an entire unit on the cell membrane here in a couple weeks. But Gram staining is a lab that will tell you about the cell membrane of a bacteria cell. To do Gram staining, you would fix the bacteria onto a microscope slide, meaning you'd have a piece of glass, you'd smear your bacteria cells on top, and then you would use heat to get them to stick onto the slide. Once the cells are stuck onto the slide, you would apply a purple stain called crystal violet, and that purple stain would go all across the cells. You would then apply another stain called iodine, and at that point the cells still seem to appear all purplish from that crystal violet stain. After that, you would use alcohol to wash off the cells, and when the cells get washed off, if they are Gram positive, the cells remain purple. But if they are Gram negative, when you wash them with alcohol, all of the stain gets washed away and the cells are left to appear colorless, which is like clear, meaning there's no stain there. Under a microscope, it's extremely hard to see things that are clearish, so we always stain specimens that we put under the microscope, otherwise you wouldn't really see anything. So the final step of the Gram stain procedure is to apply a pink stain called safranin. If the cells are purple from the original stain, that pink stain essentially does nothing. But if the cells are clear because they did not get stained with the crystal violet, then at the end of adding the safranin, those cells would appear pink. You can see off to the side, there's some real pictures of what the cells would look like, whether they are purple or whether they are pink. The color of the cell then allows us to determine whether the cell is Gram positive or Gram negative. The purple colored cells are positive and the pink colored cells are negative. So what this tells us, to bring us back full circle to the beginning, is a little bit about the cell membrane of the cell. Some bacteria cells have one membrane around them, and some bacteria cells have two membranes around them. So a Gram positive cell has one cell membrane around it, which is shown here by this orange layer. So that one cell membrane protects the cell and allows it to control what comes in and out. A Gram negative cell has two cell membranes, thus meaning it has like double layers controlling what goes in and out of the cell. The purple stain indicates a Gram positive cell, indicative of one cell membrane. The pink stained cells then all have two cell membranes. In terms of antibiotics and disease, cells that have only one membrane are a lot easier to kill because the drug, the antibiotic, the medicine needs to penetrate only the one layer that's protecting the cell. Whereas if a cell has two layers, the antibiotic, the medicine, the drug, whatever you want to call it, would need to penetrate first the outer layer and then also penetrate the inner layer. So it is almost like a double wall of protection for the bacteria cell. So in general, diseases that are caused by gram-negative bacteria are a little bit harder to treat than those diseases caused by gram-positive bacteria.